Hi, everyone. Today, we are very glad to have Dr. Prathyusha from Georgia Institute of Technology to give as our speaker. She will give a talk about the collective dynamics of active polymers from clusters to tangles. Yeah, we are very glad. Look forward to it.
Thank you, Hazen, for the introduction, and thank you for the invitation for this opportunity. Today, I will be talking about collective dynamics of active polymers from cluster to tangles.
Excuse me, can you share again? So it seems that we cannot see.
Oh, I see. Sorry, I'm just trying to. Okay, so active matter systems come across different length scales, from biomolecules to bacteria to large-scale animals, and they can occur in both synthetic system and also natural environment. So they can basically take energy from the environment and use that for useful work. So when you consider the system in collective, they can actually have various interesting emerging properties. They can form patterns, they can tangle together, they can cluster together, and also form, like, we can program the principles of this active matter to create robotic structures. So in nature, there are so many flexible active matter systems. One is basically from viruses, which can roll along the motors on carpet. And there are DNA or RNA during DNA transcription processes, and ciliates and various other organisms, bacteria, worms, vertebrates like snakes. So all of these systems, they have a intrinsic flexibility, and they have a conformational degrees of freedom. So that, and also they have activity, which can propel or do some break the detailed balance. So these activity and flexibility has emerging properties when in collection. So we can also find various synthetic flexible active system. For instance, catalytic colloid linked to linked like a chain, or a hexbug, which can also connected connected through a rubber band, can show beating like cilia-like beating, and maybe a chiral active granular chiral active disk on a vibrated channel, and if they connect with linger, they can also exhibit self-wrapping. So when we talk about active filaments confined to 2D surface, the most of the initial experiments were based on microtubules or actin filaments on motility assays. So motility assays are like elegant model system to study cytoskeletal filaments in vitro. So what do they do is you basically graft motoproteins on the glass surface, and you put actin filaments on it, and then they can convert, the motoprotein can convert ATP to ADP, so that actually eventually creates a self-propulsion for the filament. So these are, like, for example, two actin filaments or microtubules on motilities, and on the right one we have this bacteria gliding on surfaces. They also propel along the chain. So we were initially interested in the pattern formation of collective flexible filaments. So to begin with, the experiment which we are inspired by was, like, if you put many of the actin filaments or microtubules, they exhibit various pattern formation, and that can have a very biological relevance. So inspired by these experiments, I tried to do a modeling of self-propulsion active filament, where we were interested in the self-propelling filaments, where it can move along the contour of the chain. There were initially few modeling aspect where you have on the left side, you have a model where you have connected rod and bead spring model where there is no flexibility, but they can propel along the chain. So that shows different dynamics like rotation and then snaking-like motion or translation when you change the stiffness. But then later on, there are bead spring model for active with a finite stiffness, where you have the active force is along the bond between two beads. So that can actually show various intrinsic or conformational changes even in a single filament level. So for example, in this case, you can actually see that when you have a smaller kappa and propulsion, they can have a snake-like motion where every filament will follow each other, every bond will follow each other, and it move like a snake. And then another interesting property is that they can actually bind or coil and uncoil. So that also shows very interesting property when in collection. And finally, they can actually spiral around and collapse to like a diffusive spiral. So inspired by that, we actually created a model for active filament where the activity is along the tangent, but it is actually a combination of two neighboring tangents. So which also makes them follow the along the contour. So this is actually work done in collaboration with the Rasco and Silke. So we have many of these filaments collected together. They are all steric repulsive force filaments where there is no overlap between them, and we consider them in 2D, and we have a bending potential and a bonded interaction between two of them. And we can immediately think of two dimensionless number, which one is the persistent length divided by the length, which is basically a thermal dimensionless number for a passive polymer, and a Peclet number, which actually defines the convection to diffusion. So these two defines, by tuning these two parameters, we can get different phases. So we solve this Langevin equation. And well, let's see what we get. So we observe various non-equilibrium phases. So we also look at the very highly dense active polymers we kept the density around 0.65, which is very close to the jamming transition. But at very small value of activity, the system behave like a passive polymer melt. But then when we introduce the activity, the system start to flow. So that we call as a flowing melt. So initially I told that there can be at intermediate persistent length and also the Peclet number, the polymer can wind and coil and uncoil. That actually generate a swirling-like motion where individual filaments basically open up and close or coil and uncoil. That create a flow internally. And then when you increase the stiffness, these filaments come together, and then steric repulsion between them align them, and they can actually cluster them together and propel together. So at the end, if you have larger propulsion, it can create spirals. So individual polymer can actually wind up to coil. So it feels like they are diffusive spirals, rotating spirals. So,
Excuse me, in the last slides, what does the color mean here? The darkness.
Color means they are different. I mean, individual colors. Like every polymer is colored differently. That doesn't mean anything.
Okay.
Just to show that they are mixing, and if you start with a gradient of colors, it shows that it is mixing. So rather than yeah. So we study the center of mass motion of the active polymer. So how this system actually move in the collection. So we take the average center of mass of each individual filament and calculate the average center of mass diffusion. So in the flowing melt, which is like a very highly dense system, so it behave like a subdiffusive motion even at long time, because that is very relevant to very similar to super cool liquids. And then if you increase the activity, if you introduce the activity, that system try to flow, and that at long time we see a diffusive behavior. And in the phase segregated state, where you see this bundle formation or stiff filament regime, where you see ballistic regime to a super diffusion, and then at long time, probably a larger Peclet number, you don't even actually recreate, regard the, we won't actually see a diffusive behavior. And in a larger Peclet number regime, so after initial ballistic regime, it actually turns to a diffusive regime, because when they form a spiral, they diffuse around and around the center, and they never get out of the spiraling state. So we can also calculate the diffusion coefficient and the larger with the different Peclet number and also the different stiffness. We see most of the cases this is diffusive, but there are when the system become largely phase segregated and large clusters, we don't even see a non-diffusive regime. At low Peclet number, we also see a linear dependence, which means that the system actually the activity actually can induce, it increases with the activity, so the diffusion coefficient increases linearly with time. So as I said, we see that there are new ordering. So when these are not binary collision, there are many filaments which actually come together and then they collide with each other. So since we have steric repulsion and then these filaments are polar, there is a directionality. So we can basically calculate the nematic and polar ordering. When two filaments come together, they will try to align, the steric repulsion will try to align them. So we get basically nematic ordering. And since there is a directionality between the filaments, like the two filaments, whether they are going in one direction or two direction, we can also calculate the polar orientational order throughout the system. So we see that the intermediate regime where both nematic and polar ordering is higher, which means that in the for the stiff filament, where they try to align together and they also propel together. And in nematic ordering at the low values of Peclet number, meaning that they behave like a polar melt, where it doesn't, even though the steric repulsion is playing a role which aligns them set to each other. So that gives this ordering. So finally, we can basically create a phase diagram. So this is on the left, you see a single polymer phase diagram where the Peclet number and the stiffness is of the persistent length, or the dimensionless persistent length to length, and also Peclet number. So you see that there are various regime, like as I said. So at low values of Peclet number, you see a polymer regime. And then there is a weak spiral regime, which happens where you have coiling and uncoiling of the filaments, and a strong spiral regime where the filaments get trapped in these spirals forever or throughout the simulation. So we can basically create the same thing for this highly dense active filament system. I would want to make a point here that if you change the density of the filament, this phase diagram will change. You will see different other phases, but overall the phase diagram might look like this. You see a melt, and then it actually increases when you increase the Peclet number, you see a flowing melt, and then you create swirls, spirals, and a phase segregated state at this. So now I change to a different topic, where when you have so in reality, in active and passive mixtures occur in environment. For example, actually active systems, not just in environment, there are many other passive particle, and many, for example, in a system where you may see some debris, food, all of the system which are passive in nature. So active and passive mixtures are also important. So we try to see how an active and passive particle mixture behave, and then how the flexibility of active system can actually study. We can study active filament dynamics in the system. So to begin with, so if you have a bacterial system and if you put a polystyrene beads, you see that passive polystyrene bead, this activity of the bacteria can actually enhances the diffusivity of the passive beads. And we also see that if you put bacterium or passive colloids in mixture, you can show that they can exhibit clustering behavior. So, so how the flexible active agent interact with passive environment. So now in this system, the biological system which I look at is a slightly more macro scale, which is a California blackworm, which is like 0.5 millimeter in 0.3 to 0.5 millimeter in thickness and 10 to 50 millimeter in length. So how these systems actually interact with the dispersed particle. For example, here, this is an experiment done by Hari, and this work is in collaboration with Hari and Saad. And if you put bunch of these filaments together and put an algae, so they can collect or clean the environment or clean the system. So this show that they can aggregate these California blackworms can aggregate these particles. So this is also more or less influenced by the mucus, which is aroused by this worms. They can actually secreted by the worms. They can collect these algae. So meaning that if you think about a physical system, there is an active filament and actually have an attractive mucus between attractive interaction between this passive system. So this is
Prathyusha, quick question about the algae. Do the worm eat the algae, or are
I
they?
think some, but not in big.
Okay, and the algae by themselves do not cluster, but form sort of loose fractally.
No.
Okay, so but they're alive. But the algae is alive.
But again, if it is in large collection, probably yes.
Okay.
But otherwise, I think they are dispersed.
And is that something that the worms encounter in the natural environment?
Yes.
Okay.
Yeah, and then they are also so now so Harry did another controlled experiment in a lab where he put a bunch of sand particles and then see they are more or less they don't get attracted to sand particles. So you see that these dispersed particle get actually clustered by the motion of the active filament. So inspired by this, we can do some simulations. So currently there are very few active polymer and passive particle mixture simulations available, where one of them actually talk about how the dynamics of the worm changes rather than how how does that affect the system. And I also looked at how the transversely propelling polymer can actually be used to sweep the uh, thermal passive particles. So what about a system where they are not thermal, they are passive particle which are stationary like sand, and they only move when you push them with a polymer. And at the boundary, we have which mimics the actual system where you have boundary particle which are stationary, but they are also repulsive. So, and also the grooves at the boundary actually make the polymer to get reflected back from the boundary. Otherwise, it spends more time at the boundary. So it creates that much more particles can actually, go inside the arena of the system rather than spending more time at the boundary. And then we use the same active polymer for 2D, and we solve these overdamped equations of motion. And then you see that as a function of time, we start from a very dispersed particles, and then during the motion, it can actually collect or aggregate, cluster these passive particles. So these are like, so we also look at how the flexibility, like what we change the stiffness of the filament, whether that actually changes it, whether it makes it better. So we see that when you have flexible filaments, it helps them aggregate rather than the stiff filaments. So now how do we identify these clusters? So we have an in-house calculator code to analyze it. So we have, like, for example, this is on the left, you have dispersed particle, and then you can identify the cluster within the interaction regime, and then you identify these numbers. And then there are two numbers which are important in the rest of the talk, where you have total number of cluster, which measures the number of clusters in the system, and then average cluster is cluster size, which is S, which is basically we can which tells the number of cluster size, the total number of clusters, sum of the cluster sizes divided by the total number of cluster. So that actually tells us whether how many clusters are on average in the system. So we also looked at different lengths of the filament. So we see that, so on the left is you start from a larger number of particles, where I take 500 passive particles, and they are very dispersed, and then to begin with they are all single cluster of size 1, and you see that they actually go down. The number of cluster goes down on an average, and that number and the decrease also, I mean, longer the filament, you get more clustering, which means that the longer filament favors the clustering, and the short filament actually decreases the less number of filaments, the less number of particles. So we also measured the average cluster size in all the systems, and also they change the stiffness. So we also see that the longer and flexible chain favors the clustering. So now we look at, by getting this inspiration from the biological system and also simulation, can we actually create robots made of polymers, which can be connected like a long chain? Can they also behave similarly? So this is also work in collaboration with the Rosa, Elam, and Antoine in University of Amsterdam. They have these hex bugs, they connected by rubber bands, and they have these passive particles which are also stationary, which are like sand particles. They don't move by themselves, but when the activity is induced on them by the robot, they can actually behave very similar. So we have actually, so this is a plot which actually tells across scales and across different systems. Most of the cases, the system behave very similarly. In biological example, worms and robots and simulation, we see very similar behavior for the average cluster size as a flexibility and the length. So we try to say that the flexibility and activity of the system is important, and we can actually use, and this is very robust in every system. So this will be coming out soon, and hopefully in a few months. And now I switch to clusters in active system, where you have it's compared to maybe passive condensed matter system, where you need an effective attraction between particle to form these domains. But in a active system, even in the absence of any attractive interaction, they can form these domains. They can form colloids, for example, active colloids with long range interaction. They can form droplets-like structure, bacteria, or light-induced active colloids. Even in ants, they can form these domains which make them together. So when it comes to the flexible active system, the topological complexity increases with the aspect ratio. So for instance, if you have passive and also active system, when you have a shorter filament, they form less less complex structures. When it becomes longer filaments, when in the collection, they form this highly hierarchical topological complexity, which is difficult to actually break. So, so now I take you to the fascinating worm collective. So these are the California worms. So right now, the previous previous slide, we talk about how the worms interact with the other passive particle. Now I will be talking about how these worms actually interact with their own entities. So if you put many of them together, they can basically form this blob. They can entangle with each other and create this blob. And these blobs have very fascinating rheological property. They can behave like a fluid or behave like a solid. They can also sense hot and cold temperature, and they can also tangle and entangle in ultra fast way. So another interesting behavior which we see for the active worms is the phase separation. So they can also create, if you start from a very random dispersed worms, they can also behave like a active, they can also form domain. They can club together, they can form this domains, and these domains can grow and then form structure. So this is an experiment by Endurance group, and this is basically on tubifex worms, and the right hand side is California worms. They are very similar in nature, except that the California worms are much more ballistic than the tubifex worms. And so to model such system, so we do a bottom-up approach where you have to think that, okay, they have, what is the individual dynamics of a single worm? So we can see that they have this self-propulsion, and also they turn around their head very often. So that actually can change the dynamics rather than just self-propulsion alone. And so we create a model where you have an active head, which has a propulsive direction, which has an angle with respect to the x-axis that turn around. And then, for example, this vector can turn around left or right based on a exponentially distributed noise, exponentially distribution, and it can change +1 or -1, which means it can go clockwise or anti-clockwise to form this structure. So, and then we have this propulsive force and then the angular velocity that actually together will the tangling dynamics is decided by this R vector R and also the gamma. Gamma is the inverse of the exponential distributed time. So you can basically have different dynamics based on this R and gamma. So if you think about a chiral object, you see that if you don't have any connective change, this R represents this behave like a chiral active particle, and the R represent the radius it can actually make when without any noise. So this is basically the different dynamics which we see in our system for a single filament. You see that larger the R and small the gamma, it can behave, which can more turn around, and smaller gamma means it turn around faster and moves propulsively. So now we look at the collection of active blobs. So if you put many of these filaments together, what happens? So on the left, you see these California worms. We can form tangles together by braiding with each other. And similarly, if you put them, in simulations, if you put long chains with this tangling dynamics, they can behave, they can form this. So now, like we saw that there are many domains. Domains can be formed in active ones. So we look at whether the actual model can also behave similarly. So if you have like a larger system, many filaments together, and if you put them together, they form this domain-like structure, where you see that they can basically connect with each other, tangle with each other, and form these domains. And you see that these are also like these domains can also fuse with each other. You see, and one thing to note here is that there is no hydrodynamics here, and also in experiments we see that the worms are also not hydrodynamically driven. And until unless they meet, they don't tangle with each other. If it is in a dilute system, it's very hard to actually make clusters until they meet each other. If you take a larger system, but if they are nearby, then they try to form tangles.
Prathyusha,
Mm.
the simulations are in 3D or in sort of quasi 2D confinement?
It's a quasi 2D confined system.
Okay. How much, and so when you're saying tangled, so there's enough height that the polymer can go on top of another to wrap around?
Yes. It's not like it too small, I would say. Let's say half the polymer size.
I see.
Or,
I see.
or along is Z, actually.
In Z direction.
Yeah,
And for the experiments, have Harry and Saad tried to change the confinement in Z?
Okay, so in confinement in C, meaning like these are like worms in water.
Mm.
So if you change the water thickness, they can also, there is another phenomena called worm buoy. They can actually attach their tail to the top, and it may not see it may see a different behavior. So you need to have enough water to, see these blobs.
I see. And over here, that the time scale of the experimental video is that's real time. Is that correct?
Yeah, this is real time.
And so the course, so presumably will you show us something on coarsening and the temporal dynamics of how these clusters form and they coarsen?
Yeah, I will show you. This is bit of an ongoing thing. So I will show you what I have, and maybe in few slides. Yeah.
Yeah. And I think Daniel had a question.
Yeah.
Which was whether these results are sensitive to the choice of making only the head bead active. Do you not get these clusters if you have uniform activity along the chain?
For example, if you have a uniform activity, just the 2D model which I was describing, I don't think that they will form. They can form like tangles, or like they will form like a interpenetrating filaments, but not like these tangles, because I mean, if you increase the dimension, they may form like, they will overlap with each other, or not overlap in Z, but not form. I don't think that they will form these tangles, entanglement per se. Entanglement, of course, but tangles, I'm not sure whether they will have this tendency to form these structures. I'm not sure.
Okay. Thank you.
It also looks like you're using that combination of parameters that you showed, I forget, R and gamma, which so that these do not propel away too much. Is that the idea here? Because
yeah, I mean, there are cases when even if you propel them, even when they propel, they can also meet and tangle if you are in a right parameter space. I agree that they don't propel much, but of course they can also meet and form tangles. But maybe that structures are not very stable, because they can still form, there is no effective attraction between them unless they move. There is no attraction, so they are also repulsive.
okay, thanks.
Can I ask more about the biological? Do they have some type of communication, the worms, between them? Is the model, I assume, ignoring that, or is it like a state that
I think there may be something touch. Touch is something, maybe a sensing thing, because otherwise they don't. I don't know more about biological aspect, but whatever I know is like they don't interact until they meet each other, and once they are very close by, they try to form these bundles or blobs.
Do you know why they do it?
So there are various aspects, like one is like they want to stay away from the predators, and also to reduce the evaporation, etc.
Okay.
Okay, so how they form these domains, where you have this entangled blobs. So they will tangle to. So you see that when they are close by, they will fuse together by forming entanglement. So even in a dispersed state, where when they are close by, they form these blobs and they connect each other by tangling, and they can also form these blobs together. So one aspect in all these systems are how do we identify whether they are forming domains or non-domains. I said that they are also topologically connected. We can basically calculate the linking number, pairwise linking number, which is a measure how many of them are connected together within the block. So in untangling, so you can also tune the parameter where it actually form, they can propel and untangle, and there are cases where they can form tangles. So you see that the blobbing have the linking number is higher for blob cases, which is and one thing to be noted, all these systems are open curves in the mathematical topological aspect, and they are not connected at the ends. And if you are untangling them, when during the untangling case, the linking number is close to 0 or 0 because there's no connection. And then we create a phase diagram based on the propulsive force and the angular velocity. So at larger and depending on the phi value. So we also see that at a smaller value of R, we don't see much of clustering, and larger R, we see the clustering. And also depends on the initial density. So if you have a very dilute system, then you will not see this clustering, but in a larger case, you see the clustering. So in summary, I want to tell you that I have shown clusters of active polymers in a large dense system. And these systems can also cluster passive particles, and they form blobs, and then they can also form these domains. With that, I would like to acknowledge my collaborators. So the initial part of the work, I started with Rasco and Silky, and I was a postdoc in their group, and currently I'm in SAS group, and I am collaborating with Harry, who is recently graduated, and also the robotic aspects. I'm collaborating with Intuan, Rosa, and Ella, and NIH for funding. Thank you. Hello.
Oh, okay. So now questions.
Hi, can I ask a question?
Yeah, of course.
Yeah, hello Prathyusha, great talk. I was wondering about the circular the circular topology one, where you have active polymer and then passive things, and they're forming clusters. Is there I saw in the boundary there are like some extra beats. So what are the frequency of that, and why do you have that on the circular boundary?
Oh, okay. So that is basically for the polymer not to get stuck at the boundary. For example, if you have any polymer, it can actually reach the boundary, and they can circle around the boundary. So these bumps, three numbers, that 3 beats form these bumps. They can actually make them reflect back, so that it will have more particle inside coming towards the arena rather than getting stuck at the boundary. And we also did a test on how many. So this is basically replicating the experiment from advanced group. Like we also see that there is no difference. We have a frequency which is, I think currently I do some 30, but we can also vary it, and it doesn't change the dynamics quietly.
Okay, thank
But
you.
if you have larger number of things, that then it behaves like a boundary itself. So you need to have something in between. So,
That makes sense. Thank you.
Yeah.
right.
How's it going there? Really beautiful talk. I have a question about the typical size of the clusters. Is it possible to tune that with the length of the polymers?
Yes. So
and
it
the
actually depends
activity?
on the length, basically.
Mhm,
So
yes.
again, if you have a shorter filament, I think you will not form this very large clusters because, and also they also have difficulty in tangling with each other.
Yes.
The size of the polymer defines the cluster size and also the density.
Yeah, I was going to say, if you go to, let's say, somewhat shorter, not much shorter, but that they can just entangle every now and then also escape every now and then I think you could get a very rich parameter space with velocity and everything and breakout events and all of that. So yeah, it seems really cool.
Thank you.
I see we do have another question from the chat. Oh, hi, Tim, are you about to read it?
Oh, yeah, I'm about to read it. So a question from Richard Gordon. Tony Wasby showed that certain algae in a 3D flask would contract MSC, keeping the shape of the flask. Have you seen this behavior?
No, I don't think we looked at such a large density because we also looked at how they, I mean, this is also an observation Hari actually found, and then we tried to analyze it, and it's also like how they clean the system. For example, if you have like a lot of dust or debris, so how the worms actually clean it, like they do clean, but this maybe you need a very large collection of algae. Yeah.
So yeah, sorry, go on, Haisen.
I have a question about the dynamics of the growth of the cluster. So have you have some measurement like how the size, like the n inside the cluster over time, if there is some
so
exponent?
I have done some analysis on that. It actually goes as a t to the power 1/3. So we are still trying to figure out why that happens for an active system and an active polymer case. So yeah.
Okay, maybe I lost my question now. So thank you for the talk, Prathyusha. I mean, it was really, really very beautiful. For the part that you just mentioned with regard to cleaning and clearing of passive particles, the simulations that you showed, if I remember, they all primarily involved single active polymer within a bed of some passive stationary obstacles that you could move. And I wonder if there's interesting questions that you'll have looked at on optimizing cleanup in terms of both, not just with one particle and playing with how active it should be and how flexible it should be to clean up an arena of some given area in the shortest amount of time. Or maybe you do the least amount of work to do the cleanup. But also if you have many particles, many filaments, because there's a different limit in which, if you had many of these sweepers and they all are long, and if you have many of them, then they will start interacting and they will typically tend to line up if it's all confined in 2D, so as you've already shown. And then you will get a passive, you'll get a sort of active nematic like phase, which is sweeping up a passive density. And there you cannot clean up because the density will always form clumps. And will never, it'll form patchy clumps and it will never get aggregated into one cluster, and you will. So there might be an interesting sort of a tragedy of the commons there, where you have too many sweepers and then sweeping polymers, and you fail. And if you have one, then you do sweep, but you take long time. So is there an interesting trade-off?
No, I think that is like, since we know that if you put one filament, I agree with you that if you put many of them, they may not be good at cleaning. So we looked at how we can actually use them for cleaning rather than, so we didn't look at the limit at which how many of them actually can, better at cleaning. Like if I put one, I agree with you that they can also align with each other and form like they can move together. But how do they, like, I don't know the numbers, but because we haven't looked at the number of or the limit of the system. So yeah, that's an interesting question. And yeah.
Okay, thank you.
I wanted to ask you, when you first introduced the passive particles in the single filament, it looked like there might have been a stable orbit where the filament or the worm had moved enough passive particles out of the way that it now had a clear path and it was bouncing around screensaver-like. Is that a generic outcome?
Yeah, so that is actually another thing which I didn't show up. So because there is a W, like if you want the polymer, actually there is a clear path, and also so there is a distance between this the way the polymer sweeps it, so that has a width. So we are actually doing some analysis more than the flexibility or stiffness, so that width is also something very interesting, and that actually determines how good your cleaning is. Yeah. So I haven't shown that, but we did that analysis, and thank you.
Okay, thanks.
Thanks for the fascinating talk. So actually, I could ask multiple questions, but in the interest of time, and I'll just ask one. So I'm really interested in the maybe it's a question more for your experimental collaborators who did the chain of hex bugs.
Yeah.
But maybe do you know, so in my experience, these hex bugs often have an inherent chirality because they're not weighted uniformly. So if you just let a single hex bug go, it'll try to make circles. So do you know how your experimental collaborators circumvented this issue? Because to compare with the worms or the model, you probably want something achiral. And also, how do you vary their bending stiffness in the experiment? Or you just make them longer or shorter?
I think they also have they have a rubber connected linger, so that with varying the thickness and width, I think they can change the bending stiffness. But when you put them connected, they form the self-propulsion, which is similar to a self-propelling polymer. But again, like if you change the chirality, I mean, if you change the stiffness, they can also go in a circle. So I mean, it's the stiffness. So yeah.
Okay. So you have to keep the stiffness lower to make it achiral, or?
Yes. Yeah, I mean, there can be some tricks which I don't know more in detail, but I think
we look forward to the method section of your paper when it comes.
Yes. I mean, hopefully it will come out soon. Yeah.
All right. Thank you so much.
Thank you.
I'll tag Suraj, who has his hand up.
Sorry, that was from before.
So I have a question about the final phase diagram that you show about the entangled phase and the entangled phase. I want to know that whether that's a phase diagram for starting from the same thing, and then the entangled phase that they can form in entanglement, and the other phase is that they cannot form entanglement, or that the entangled phase means that starting from the entangled phase, they can entangled from that.
Oh, okay. So that is basically from a, so I start from all of them start from an entangled phase, dispersed state, and then whether they can form tangles. So that's what I look at. But again, even if I start from a bundle state, and if my parameters are varied, they will go back to in long time. It will show what it actually look like.
So you mean
what I'm trying to say is that even if I see a domain or phase a separated state, and if I tune the parameter, it can entangle.
It can entangle, and that phase diagram does not change with the initial condition.
No.
Okay. Any other questions?
I noticed you use Langevin dynamics with momentum term, right? Not many people in the field use Brownian dynamics. I was wondering if you could comment on what are the consequences of that choice?
Okay, I think this is one of our referees also asked the same question.
It wasn't me. It wasn't me.
Yeah. So that is only in the the first work which I was talking about, the clustering of actin filaments. So we also answer that in our appendix that so in long time scale, of course, initial collisions probably this the having the momentum term or having the mass term, initial term will affect, but in long time they don't see.
Okay.
Yeah, so long time behavior, we see similar MSDs and similar systems. So it's not affected long time. So of course it affects at short time, but not maybe collision might have some impact, but long time behavior is similar. Yeah.
Okay, makes sense. And do I understand from your answer then that for the more recent projects, you've switched to Brownian dynamics?
Yes. Yeah. So
Okay.
that's much more for a, so I'm mimicking worm, they are more underdamped. So I use those questions.
More underdamped or more overdamped?
Overdamped, sorry. Yeah.
Oh, okay, okay.
Yes.
Got it. Okay. Could the, what do I want to say? Oh, the self-intersection question. When a worm intersects itself, is it the same interactions as beads on different chains, or does it freely go over itself?
Sorry, I didn't get your question. Like when you form the spirals, you mean?
Yeah. Sorry, in the latter part of the talk, when you went to 3D, it still self-repels when it meets itself?
Yes. There is no attraction. So throughout the system, there is no attraction, effective attraction. But then the steric repulsion can also kind of act like an attraction. If you put two of them, two filament, they can actually get stuck. So, but not an effective attraction or attraction in per se.
Well, but I meant when the same filament wraps around and hits itself, that's also a steric repulsion?
Yes.
Okay.
Yeah.
Thank you.
So, I mean, again, there are more interesting aspect of this model. They can also form knots and all sorts of things. So, which is maybe interesting. Maybe we'll have more. Will come out maybe soon, maybe sometime.
Okay. I look forward to that.
Active polymer knots, so they can wind around, and then because they have this is the dimension, so they can wind around and form this knot, and the knot can diffuse towards the tail, and then unknot by itself. So.
Very cool.
Okay, if there are no more questions, I'll end the recording now. Oh, there is one more. Okay, yeah, please continue.
So this question is not for Prathyusha. This question is just somebody asked a question about chirality. Just I want to know what chirality means in this aspect. Is the time reversal symmetry is broken, or in what context it is chiral? Does anybody, not necessarily Prathyusha, just in a general question.
So I think that is in terms of activity, whether they can, by the way, that is my husband. So he's a condensed matter physicist, and he comes from a different chirality or chiral system. So yeah.
So should I answer it, because you can answer him later?
Yeah, of course.
So
Yes,
I mean,
please.
in my question, I just simply meant like these hex bugs, or there are other active particles like bacteria, they kind of move in circles, and the circles could be preferentially clockwise or counterclockwise. So it's a left-right symmetry breaking. In the hex bugs, it probably happens because the weight distribution is not symmetric.
Okay.
So they kind of move in counterclockwise circles. That's
Okay, it's motion related left and right asymmetry. Okay.
Yeah.
Okay. Thank you.
Okay. Thank you, Prathyusha, for the wonderful talk. And I'll stop the recording now.